All right, our fourth speaker today is Dustin Wolkis, who is the scientific curator of seed conservation at the National Tropical Botanical Garden, where he serves as the principal investigator for seed conservation research. Dustin is especially interested in seed longevity and wild species that exhibit anomalous responses to conventional seed storage methods. Today, he'll present on applied seed conservation biology to support seed banking.
Aloha, everyone. It's a real pleasure to be here. I did my PhD at the National Natural History Museum of Denmark, so it feels really good to be speaking here and discussing seed banking with you all today. So let's go ahead and get started. So this is an image of Kalalau Valley. It is the most species diverse valley in all of Hawaii. And I'm using this as a metaphor because the topics of this presentation is also quite diverse. And I've, of course, included some species of native Hawaiian seeds because this is a presentation about seed biology. So let's begin with this children's tale. It's called The Lorax, written by Dr. Seuss over 50 years ago. And if you're unfamiliar with this tale, the main character is called the Onceler, and he comes to this intact, pristine ecosystem. He then goes on to remove the foundational forestry from the ecosystem, and then the ecosystem collapses. However, thankfully, seed banking was practiced. And so I'll give you a little preview. The story ends, this is the kind of the beginning of the last page. So Ketch calls the Onceler, he lets something fall. It's a truffula seed. It's the last one of all. You're in charge of the last of the truffula seeds. And then we'll check back in with the Lorax and Dr. Seuss by the end of this presentation. Unfortunately, plant extinctions are not restricted to children's tales. In fact, they're happening the world over and they're accelerating. Interestingly, yet sadly, Hawaii stands out as having the most recorded plant extinctions. And just to give you an idea of where Hawaii is, it is the most remote island group in the world, about 4,000 kilometers to the nearest continent. And while we're known for our scenic landscapes and our beautiful beaches, we're also known for our flora. So we have a relatively small yet intact flora, and we're actually outnumbered. We have more species of naturalized than we do natives. And although that we have less than 1% of the land mass of the rest of the United States, we have over half of all the plant species listed on the federal endangered species list. We also have 90% vascular plant endemism. So 90% of the plant species that occur native in Hawaii occur nowhere else in the world. This map shows the number of native and single island endemic, or SIE, species per island. And I'll bring your attention to Kauai, which has the largest number of single island endemic species. And so luckily there are strategies. There's the global strategy for plant conservation in Hawaii that was regionally adapted to the Hawaii strategy for plant conservation. And in the US, there's the national seed strategy. And targets 8 in both the Hawaii and global strategy suggests preserving at least 75% of threatened plant species in XC2 collections. And this target has been nicknamed banking on nature, because seed banking is the most cost-efficient and low-risk means of housing an XC2 plant conservation collection. But what do I mean when I say seed banks? Well, you could have a natural seed bank, for example, an aerial or a canopy seed bank that's something like a coniferous forest. More likely, if we're talking about a natural seed bank, we're talking about the soil seed bank. And then you can have an XC2 conservation seed bank. And so for this presentation, when I say seed bank, this is what I'm talking about. And Colin brought up and his crop wild relatives about how there's been some genetic bottlenecks in crop species. And this is true, right? Because they've been bred to be very uniform in whatever they flavor profiles or whatever. And in wild species, it's our job to collect and maintain whatever genetic diversity remains. However, this means that interpreting patterns from wild plant species can oftentimes be challenging. And so we'll talk about some of the major challenges in seed conservation. This is having no or too few seeds, storage behavior, longevity, germination. Now, if you think that these look familiar, I would say that you're right, because these are these exceptionality factors that have been described for exceptional species. And so exceptionality factors describe challenges in the ex situ conservation pipeline. So for example, in the first step, you have to be able to collect seeds, that's EF1. And then in EF2, this is desiccation tolerance. And then EF3 deals with longevity. And then EF4 is about germination. And so we'll be talking about all of these today, except for the first one. Okay, so let's first talk about storage behavior. So this is a diagram of the structural states phase diagram of water in seeds. So we have our water content on the x-axis and our temperature on the y. And here's our happy little seed that we harvest off the mother plant in the ambient environment. And if we were to just stick this in a freezer, we would be creating ice crystals in the cytoplasm. And so we don't do that. Instead, we have our happy little seed, and we desiccate the seed first, and then we freeze the seed. And the remaining water is very tightly bound. It just kind of freezes in place and forms this viscoelastic or a glass. And this is kind of the secret to orthodox seed banking. But of course, we mentioned these exceptional species that don't fall into this class. And so the only reason we can even do seed banking is because seeds are hygroscopic. So to illustrate this, on the left here, we have a wet seed in dry air, and the blue arrows are water molecules. So you can see the seed losing water in this state. On the opposite side of that, we have a dry seed in wet air, and so the seeds are absorbing water. And in the middle here, we have the whole system in equilibrium with water freely flowing in and out of the seeds. And of course, there's a trend for both of these states to come into equilibrium. And so why do we dry seeds? Well, simply put, it extends longevity. So here we have our seed survival curves, and we have our time on the x-axis and germination on the y. And we can see that as we remove water, longevity is increased. Now, you might say, well, that's not desiccation tolerance, that's longevity. And I would say, well, you're right, but in fact, it's all four, because you would have had to get the seeds in order to do this experiment and then get them to germinate. Yes, we also show this really handy diagram, which shows the range of desiccation or drying that can be achieved with different materials and equipment. And so this is a kind of a really handy diagram for seed bank and gene bank managers. Just to give you an example of an empirical experiment, this is Caprosma cauensis, a Kauai single island endemic. And this is my former Kupu AmeriCorps intern, Emily. And we have our germination time courses here. This is time on the x-axis and proportion germinated on the y. And so she looked at seeds that were freshly collected, then seeds that were desiccated, then desiccated and frozen. And there was no difference in final germination in the fresh seeds compared to seeds that were desiccated and then frozen. So these can likely be stored by conventional methods. Okay, so let's move on now to longevity. This is typical seed survival curves, and seed survival curves inform seed bank management. So we have a kind of a flat period of time, and then a threshold where viability rapidly declines. And so it's kind of our job to understand what that looks like. And it's not only different for every species, but they're actually collection or accession level traits. And we routinely test viability to assess longevity. Recently, we looked at 295 species in the Hawaiian flora, and we came up with this regeneration or recollection interval of a 30% decline from initial or maximum viability. So in a recollection interval, you withdraw the seeds from the seed bank, you go back out to that plant or that population, if it still exists, and you recollect, thereby refreshing the seed bank collection. In a regeneration interval, it's much the same. You withdraw those seeds from the seed bank, but you propagate them in such a way that you can harvest the yield from those seeds, thereby refreshing the seed bank collection. Recollection is, of course, preferred, as artificial selection cannot act on those seeds. But of course, if the plants or the populations are extinct, then regeneration is your only option. In any case, at the time of the most recent test, 195 species had already reached their recollection interval, which means that 100 species had not yet reached their collection interval. We then took those data and made them freely available online as a Hawaii Seed Bank User's Guide, which is a nice resource for seed banks and species managers and land managers working in the state. At the NTBG Seed Bank and Lab, 21 species that that have been stored for 30 or more years, most of which are endangered, have already germinated in the seed lab. And so this gives us hope that our seed banking efforts from kind of the earliest efforts of the days are working. And that includes this Lobelia nihiauensis, which is a Hawaiian lobeliad. And this is interesting for reasons I'll get into later. But this accession represents the oldest known viable seeds of any Hawaiian lobeliad. Yes, so it's important to understand seed survival. And so there's different ways that you can do that. But one of the kind of standard ways is to use probit analysis to generate these kind of curves. And these Ki and the sigma, these are the accession or collection level traits. We also talk about how P50, or the time for viability to decrease to 50%, is a really nice metric for longevity because it's more intuitive than some of the other metrics. And then we did kind of a demonstration of how to model this in the free open software environment R. There's no papers on this. There's no YouTube videos or blogs or anything. So this is a nice demonstration of how to do just this. And we usually think about seed banks as being the only repository for viable seeds, but it's been suggested that herbaria could be overlooked repositories for viable seeds. For example, this bupleurum has germinated after 144 years at ambient herbarium conditions. And so I mentioned this to my kind of intern at the time, Susan, and she got really excited about this and wanted to do an experiment like this. So we decided to use ohia, or Hawaiian metrosideros, as our study organism because it is the foundational forest tree. So likely there'd be lots of sheets in the herbarium. And because it's so prolific, likely there'd be lots of seeds on the sheet. And also the seeds are orthodox or can be conserved using conventional methods. So we thought we'd have high success. And so we sampled from a chronosequence of herbarium specimens going back in time, several decades. And I came up with a really cool graphic to show you what happened to every single seed from every single specimen. So not one seed germinated, not one. So that was somewhat surprising given what I told you, that the seeds are orthodox in conventional conditions in conservation seed banks. However, the seeds are unlikely to persist for long periods of time in the soil seed bank. And indeed, there's no correlation between longevity in the soil seed bank and longevity in the conservation seed bank. And so you might say, oh, well, the seeds didn't survive the harsh process that herbarium specimens go through to enter the herbaria, the dryer and the freezer, but we tested for that too, and there is no significant difference in germination between the freshly collected seeds and the ones that went through the dryer and freezer. Nevertheless, it's been suggested that if you're going to do these types of experiments, that you should do them with a rare and endangered or even extinct species. So in Hawaii, we have something called the Plant Extinction Prevention Program. And this is a program that manages species with fewer than 50 individuals remaining in the wild. And at the time of this experiment, there were 257 of these species in Hawaii and 81 seed plants on the island of Kauai. And so that became our target list. And this is Kelly, my intern at the time. And so we ended up looking at 1,250 specimens, or about 86% of our target taxa. But only 11% of those actually contain seeds. And so it's unclear why that might be. It could be that botanists like to collect things in flower. More likely, it could be because all of the seeds of these rare plants were sent to propagation in the seed bank. Nevertheless, we were able to sow over 2,000 seeds from those specimens, which was about half of our target taxa. But we also observed dismal germination results. So only one seed germinated, and it was from Shedia, and it was only collected a few years before present. But then these other two events happened where the embryos just kind of hopped out of their seed coats. I should tell you, a seed is a baby plant in a box. The baby plant is the embryo, the box is the seed coat, and the lunch is the endosperm. That's a quote from one of my favorite seed heroes, Carol Baskin. Yes, and so that was odd. I had never seen it before, and so I reported it. In any case, doing these types of studies is like finding needles in a haystack, but if you don't look through the needles, you're not going to find the haystack. So we recommend that if you're going to do these types of studies, that you should do them sooner rather than later before viability falls even further. And then just recently, our herbarium curator found a jar of 30-year-old endangered species seeds that were being kept at kind of ambient herbarium conditions in this jar. And so this is Emily, and she was, of course, naturally curious if the seeds still had high viability. Well, it turned out that they did have high viability, and because they had all their provenance data with them, we accessioned them into the seed bank. And she was first author on this paper. So this is a picture of Brighamia rockii. This is kind of Ken Wood's famous picture of the species hanging off the cliffs of Molokai, the tallest sea cliffs in the world. It's also the genus that's on my shirt here today. This is one of species of Hawaiian lobelia. Let me introduce you to the rest of the family. There are 6 genera, 5 of which are endemic. And we think they originated around 13 million years ago near where the French frigate shoals are today and have radiated into 160 currently described taxa. And we have observed kind of a curious seed storage behavior from these species. And so on this graph here, we have our 6 genera on the x-axis and this measure called relative performance on the y. And if the bars are above the 0 line, then that's just kind of regular old orthodox. And if they're below the 0 line, they're desiccation tolerant, but sensitive to conventional temperatures. And so we found that in all of the genera, except for perhaps the Tremata lobelia, which is quite interesting. We also found this in Rubiaceae, Cyperaceae, and Urticaceae. And it turns out that Hawaii has the highest proportion of any flora ever assessed, exhibiting this curious kind of seed storage behavior. So Hawaii can really serve as a model in seed conservation for other tropical, subtropical, and island systems. And I will tell you that our current gene bank standards, which have been adopted by the wild species seed bank world, suggest storing seeds at -20 degrees C. And so conceptually, here's what's happening here. We have our theoretical seed survival curves here with time on our x-axis, viability on the y. And these seeds that are desiccation tolerant decline faster at -20 than they do at +5. And in the Hawaiian species, it actually takes time to detect this difference, time like 2 to 5 years. And this has been called intermediate freeze sensitive, but we'll talk about that. If we do a seed information database search for Campanulaceae globally, we see that overwhelmingly the results are orthodox with no intermediacy or recalcitrance reported. Okay, so anomalous behavior of aging at -20 C is linked to freezing and melting dynamics of triacylglycerols at storage temperature. And so that's a clue. So if we look at oil content of Campanulaceae globally, we see that overwhelmingly this species are oily, the mean of 32%. So with these 2 pieces of information in mind, we set out to use differential scanning calorimetry to determine lipid thermal fingerprints. And so the way this works is that you have kind of a sample chamber and a reference chamber. And as you change the temperature, it measures the energy output between the 2 chambers and the differences between them. So we load our seeds in, we cool the sample down, and then we warm them back up. And this has only ever been done in a handful of species, some Kufea, some orchids, neem, papaya, some citrus, and most recently, oily Brassicaceae stored in seed banks. And so we set out to do this with our Hawaiian lobelioids. You don't need to read the species that are highlighted in red, just their position in this recent phylogeny by Kagami et al. So we used 1 representative species from each clade of the Hawaiian species, plus closely related species from the South Pacific, Africa, and the Americas. And we found that overall, the standard seed lipid thermal fingerprints followed one of 2 general shapes. But first, let me break this down. So we have our a lobelia in purple at the top and a cyania in green as 2 kind of representatives. And then we have our temperature on the x-axis, and this is a warming scan, so temperature is increasing. And our y-axis is just like energy flow. So the first thing that we do is we plot a baseline that's represented here by the red dashed line. And I have the plot oriented so that endothermic events are going up. That is to say, melting events are peaks and crystallization events are troughs. And then once you do this, you can start to characterize each of these events. So for example, this is a small melting event here. These are alpha crystals. followed by alpha recrystallization. But then this is where the two curve types diverge. So in this bottom cyanea, we have one broad melting event with diverse peaks. These are beta crystals. And on the top, we have broad melting events with two distinctive peaks. That is to say, there's a crystallization event between those two peaks. Once we characterize these different events, we can quantify them. And when we do that, we find that the standard scan revealed small yet significant differences in melting and crystallizing temperatures and enthalpies. And so I'll give you just one example. We have our species on the X and our major alpha melt peak temperature on the Y. And we can see that even though those temperatures are only slightly different, they're significant. We also found that overall, the effects of storage on temperature on lipid thermal properties varied by species. Again, I'll give you just one example here. For 3 of these species, I had them stored at 3 different temperatures: the conventional -20, the +5, where we've seen increased longevity, and a novel -80 for some years. And so, for example, on the left in this Claremontia, we see that seed stored at -20 had lower enthalpy than seed stored at -80. But in the Trematolabilia on the right, we see that seed stored at -20 had higher enthalpy than seed stored at +5, with no differences in our cyane in the middle. We also found correlations between proportion germinated and lipid thermal behavior varied by species, but not storage temperature. However, this was all based on just kind of germination at the time that these curves were generated. And so what would be even better to do is use longevity metrics like the P50 I mentioned earlier as that. So more research is needed on this. I also introduced phylogeny into lipid thermal fingerprints. So this is brand new and has never been done before. And at first, when I pruned one of these mega trees, it looked like this, this trait of having the 1 or 2 curves was related to phylogeny. But in fact, when I got Kagami's phylogeny, we found actually very little support for that trait being related to phylogeny. However, the upshot is that this is new and we could use this for other traits as well. And so lipid thermal fingerprints show very distinct signatures across species that could be related to this freeze sensitivity. The vertical dashed line is our conventional storage temperature, -20, and we can see that that's kind of bisecting this big melting event. And rapid lipid thermal fingerprints offer a rapid non-destructive test to screen for potential problems with longevity. And they could be generated for all seed bank accessions. This test is actually non-destructive. And this freeze sensitivity kind of term might be a misnomer. Something like conventional temperature sensitive might be a little better, a little somewhat bulky. And just a reminder that the current gene bank standards suggest storing seeds at -20. But you know what? You can buy a -80-degree freezer right off the shelf, just like you can buy a -20-degree freezer. So it wouldn't surprise me that maybe in a couple of years, the standards change to -80. I don't know. Time will tell. Okay, let's go on to our last exceptionality factor now, dormancy and germination. So dormancy and germination, it's a little bit like studying fairy tales. Let me explain. On the left here, we have the princess and the pea. And the princess needs to have her conditions just right in order for her to get what she needs. Well, seeds are the same way. They have to have their optimal germination conditions in order for them to germinate. However, even if you have optimal germination conditions, the seeds still may not germinate. So this is like Sleeping Beauty here. Sleeping Beauty is waiting for an external signal to elicit what she needs. Seeds that are dormant are the same way. They need an external signal to elicit germination. However, if you can break dormancy and give the seeds the optimal germination conditions, then you can get nice, robust germination like Jack and the Beanstalk here. And I think this section is especially relevant for conservation and horticulture. So here we have our Brichamia rockii. This is kind of an example. Again, the species on my shirt here. And it has morphophysiological dormancy. But one of the interesting things is that these seeds will germinate in the dark. This is interesting for 2 reasons. One, it's very rare for Campanulaceae globally to be able to germinate in the dark. And 2, it could maybe in part explain some of the rarity of the species. If the seeds germinate in the dark, they're unlikely to form a persistent soil seed bank. So maybe in part could explain some of the rarity of the species. There's 10 individuals of this species left, 0 of the other one in the wild. And so we've been doing these kinds of studies in Hawaii for a while. I think that these studies are important and they need to continue. They'll make excellent student projects, I think. But now I think it's time to take a more holistic look at dormancy and germination in the Hawaiian flora and look for bigger patterns. And we also germinate seeds as part of routine seed bank management. As when seeds come in, we do something called initial viability, and then we test them through time. This is an image of ohia flowers. Ohias are native Metrosideros. And this is a species that's potentially the most ecologically and culturally important species in Hawaii. It provides habitat for birds and nectar for birds and habitat for insects and other invertebrates. And it's also a nurse log. No, not that kind of a nurse log. Oh, that is an ohia log. But this kind, log that provides habitat and substrate for the next generation of plants. But enter rapid ohia death, dun, 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 dun. Rapid ohia death is the colloquial term for two newly described species of Ceratocystis fungi, which is totally decimating large swaths of forest on the Big Island and has been detected on all of the islands. And so, as funded by the Hawaii Tourism Authority, because they recognize the threats of tourism this pose, we set out on a big kind of collecting and banking campaign. And so these 86 dots here represent collection locations across the island of Kauai. And we wanted to know if there was any correlation with any of these kind of 19 classic bioclimatic factors and seed zone. A seed zone is an area in which you can take seeds anywhere out of that zone and then put them back anywhere else in that zone with a higher likelihood that those seeds are better adapted for that area. And so when we did this, we didn't find any correlation with initial viability and any bioclimatic variable, but we did with seed zones, with seeds having lower germination in the Nepali valleys compared with the windward ranges. And it's unclear why that might be. It could be that some of the trees are drought stressed or that there's lower pollination in rainier areas. But nevertheless, the upshot here is that routinely collected seed bank data can be repurposed for other types of conservation as well. Okay, so we've talked about conserving exceptional species in a rare flora, but what about other types of germplasm? Well, we were listed as one of only 8 active fern spore banks in the world in a recent fern biology book in the conservation chapter. Certainly there are more than 8, but we were listed as one. And similar to the seed viability projects that I mentioned, we recently did this with dry land fern species. And in fact, we did find viable spores on historic herbarium specimens. And so that's exciting. And exceptional species may have bankable pollen. So there's a number of reasons why you might want to bank pollen, but banking pollen allows you to make controlled crosses across space and time. So as habitats continue to be more severely fragmented, and as flowering phenologies continue to change, it's increasingly important to bank pollen, but also ex situ. Pollen is already being shipped across institutions and able to facilitate a pedigree-based approach in meta collections. And so this is a phylogeny of a recent gap analysis of exceptional species. And then I've mapped pollen cytological type on top of it. And so pollen cytological type of most exceptional species is either not assessed or binucleate and thus presumably bankable. And so I'm going to introduce you now to 3 species. The humans are Ces and Alina, and the plants is a single island endemic hibiscus clayi and single Kauai island endemic Prochardia minor. The hibiscus just will not make seeds on its own, and the Prochardia has problems with desiccation tolerance and is short-lived at conventional conditions. Also, we recently detected coconut rhinoceros beetle on our island, which is a new threat, which we're all freaking out about. Anyway, and so we are hoping to develop a protocol for pollen conservation. And so in order to do this, we have to ask a series of quite boring questions to more interesting ones. And so the first kind of question we have to ask, because we have to get the pollen to germinate, to ask these kinds of questions is what kind of media they'll germinate in. And so, yes, so our x-axis here is our sucrose concentrations, and the y-axis in all the plots I'm going to show is our proportion germinated. And then we have our solid agar and our liquid media here in the green and brown respectively. And the first thing that we can see is that these two species are on opposite ends of the spectrum. These two species, I should tell you, have very different physiologies, as we're about to see. Yes, and so that's interesting. And then we have to pick something. So we pick 10% agar for the palm and 30 or 35 and agar for the hibiscus. Great. Let's ask some more interesting questions. It's been proposed that the pollen germination temperature is the most important trait for regeneration of a species. That's not me saying that, that's others. In any case, it's important for ex-situ conservation as well. And so here we have our temperature niche breadth for the two species. So we have our temperature now on the x-axis. And the first thing we can see is that our Pichardia has a very narrow temperature niche breadth, especially as compared to the hibiscus. And I should tell you the error bars are 95% confidence intervals from a GLM. Yes, so very different. And then we have to pick something. So we pick kind of 25 for the palm and 20 or 25 for the hibiscus. We can also look at how long it takes pollen to dry. So here we have our palm, which is dry after like an hour and a half or something. This was hydrated. And then our hibiscus clayi, still, this is right off the plant, not rehydrated, still not kind of dry until a couple hours. We can also look to see about the timing of germination. So this is Pichardia minor pollen. This is about the last 2 hours of a 3-hour time lapse that we took. Yes, pollen does germinate. These are pollen tubes. And then we can ask, I think, kind of the most exciting questions, which is about longevity in response to moisture and temperature. So here we have our Pichardia minor, and we can see that we don't have, and they were stored at these 4 different relative humidities. So we can see that we don't have great initial quality here, but nonetheless, regardless of storage temperature, the pollen's still kind of mostly alive, at least at the lower RHs after almost a month, and that maybe this kind of middle RH is good. However, if we freeze that, this is after 4 days, we can maybe extend longevity. So that's great. But now look at our x-axis and our hibiscus here, which is quite dead after just 1 day. So yeah, very different here. And then if we freeze that after just 1 hour in the storage environment, 4 days later, that did nothing for us. So that didn't help us out anyway at all. Yes, so I really think that pollen storage is the next critical tool in the plant conservation toolkit. It's been used for a very long time in agricultural species, but has yet to really take on in ex-situ conservation. So I think this is the next big thing. So let's check back in with Dr. Seuss and the Lorax. So the very last page of this story ends. So Ketch calls the Onceler. He let something fall. It's a truffula seed. It's the last one of all. You're in charge of the last of the truffula seeds, and truffula trees are what everyone needs. Plant a new truffula, treat it with care, give it clean water and feed it fresh air. Grow a forest, protect it from axes that hack. Then the Lorax and all of his friends may come back. So this is an example of ex-situ conservation efforts supporting in-situ restoration efforts. It's also a metaphor, just like seed banks, which give us hope for the future. Thank you. I have a plug here. So we're very happy for my technician, Alina, to go have her baby. That means that there's an opening. So if this kind of work looks interesting to you, please email me.
I think you're going to get a lot of hits after that. We have a question online from Kay. Thank you. Regarding the fern spore research, can you summarize the results relevant to conservation horticulture handling of fern spores?
I cannot summarize them, but I can tell you that 2 that were observed to have germinated with these Doryoptis decora and this Pteris hillebrandii. Yes, they didn't all germinate, but a lot of them did. Yeah, that was just accepted to American Fern Journal.
Thanks again.
Yeah, thank you.